Yasmin is the Ward Coleman Chair in Translational Neuroscience, as well as Professor of Psychiatry, Neuroscience and Pharmacological Sciences at the E. Kane School of Medicine in New York City. She is also Director of the Addiction Institute in the Mount Sinai Behavioral Health System. Using translational approaches which embrace animal behavior, molecular biology, cell biology, pharmacology, psychology, neuroimaging, bioinformatics, and biotechnology, she's truly pioneering in exploring the neurobiological correlates of behavior and generating insights that can be applied in advancing treatments and interventions and helping people to overcome addiction. Her work has revealed the effects of exposure to drugs early in life, whether prenatally or during adolescence, and how it increases vulnerability to addiction later in life. Focusing on the systematic study of the human brain, of drug abusers and subjects with psychiatric disorders in relation to opioid neuropeptide, cannabinoid, and dopamine neural systems, she has been a trailblazer in understanding the role of opioid peptides in drug abuse and the transgenerational effects of early cannabis exposure and the developmental brain and behavior. The importance of her work With the importance of this work, she has received many awards, including election to the highly prestigious United States National Academy of Medicine, as well as the American Society for Neuroscience, New York Academy of Sciences, and the College on Problems of Drug Dependence. Importantly, she is also a lifelong champion of underrepresented groups in science and medicine, a commitment that was recognized with the Society for Neuroscience Mike Salpeter Lifetime Achievement Award. She was also the founder director of the Diversity in Biomedical Research Community at Mount Sinai and sits on many boards and committees that aim to enhance diversity, equity, and inclusion. Her talk today is on the vulnerable brain, pathways to and from addiction.
Thanks, Martin, and to John and the program committee. It's really a true honor. It's a little surreal in part because the last time I was at the ECNP conference, I think was like probably 2 decades ago. And I feel that I'm a child of Europe because I was trained at the Karolinska Institute, so it feels good to be home. So I look forward to talking with all of you. It's an amazing program. So I look forward to attending a lot of the different sessions that I see that are really great. So today I'm going to give an overview of some of the research that we've done, not going into all the minutiae. There's a lot of unpublished data, but it's more telling you where we've come from and where we're going. And for me, I'll start off that I don't have any disclosures, except the disclosure that it takes a village to do a lot of the research that we do. So I thank all the people in our team, preclinical and clinical, and I'll emphasize some of their work as I go along. But we are all in our village trying to understand and confront addiction. And it is not just a US problem, it's a whole world problem. It's a whole world problem with a lot of different substances that are abused. This is also a challenge because of all of the issues that come with it, from the overdose deaths that we see in our society. The stigma is a huge aspect of addiction as compared to a lot of other disorders. I mean, we talk about mental health challenges, but within mental health, addiction still has this, negative stigma with it, not just for the people who suffer from these substance use disorders, but even for the clinicians who try to treat them. And indeed, a lot of the treatments that are there are either not used or not suitable for many of these substances. There's also a huge economic burden of substance use disorders, and it really has our healthcare under siege. Yet still, we are not moving far enough with translating the science that we do to actual clinical interventions. So today I'm going to talk about 2 of the substances that my team focuses a lot on right now, actually for many, many years, cannabis and opioids. Clearly, in North America, we have an opioid epidemic, and that's going around the world as well. And cannabis has come up to be very big in many different ways in a number of societies as we have legalized cannabis in many countries. And what are the consequences, not just in terms of the recreational use of these substances, but opioids obviously are an amazing medicinal tool, and we're also now trying to understand if cannabis can be one of those as well. But when I think about addiction, we think about it in different contexts. Yes, I studied the human brain, and especially obviously the adult brain, looking at a lot of molecular studies in the post-mortem human brain, because many people with substance use disorders pass away. But it's about addiction risk. It was one of the questions that I had early on when we're studying our adult population. And part of the story I will tell you is, for me, addiction risk has a developmental origin. But of course, the environment makes a huge impact on that developmental trajectory. We're not going to, discount genetics, which does play a role. I'm not going to go into that today. But together with the environment and the genetics, we know these epigenetic, I would say, tags that really take on what has happened during our early life to interact with who we are in terms of our genetic blueprint for that adult behavior. And those behaviors really are what we think about when we talk about risk. We have comorbidity with many psychiatric disorders when we talk about substance use. So it's a very complex disorder, but of course, it has to start with the drug exposure. So as I said, when we think about addiction risk, and in fact psychiatric risk, we often study the adult. But one of the things for us I don't know if I want to say very early on, but it became clear that there's a developmental origin of these mental health disorders. And often we look at adolescence, but even into as early as the prenatal time period, that's very important for later adult behavior and vulnerability. What I'm going to talk to you a little bit about today is the impact of cannabis on those developmental processes. And it started many years ago in studying the endocannabinoid system, where THC, the main psychoactive cannabinoid in the cannabis plant, binds. And the cannabinoid receptor, the CB1 receptor, is the most abundant G-protein coupled receptor in the adult brain. Throughout the cortex, basal ganglia, amygdala, hippocampus, cerebellum, it is everywhere. It's really key to modulating neuronal function. But we started to study the human fetal brain, and the expression of the cannabinoid receptor is different, and we're not surprised by that. And in particular, we could see it in mesolimbic systems at this development stage, mid-gestational stage in the human fetal brain, in the amygdala, in the nucleus accumbens. And it really, many people in this field has looked at the endocannabinoid system during development, and we know that it's critical for neurodevelopmental processes, from hardwiring of the brain to, making of new cells, whether neurons or glia, their migration of these cells, the synapses that are formed, and so on, even into adolescence and young adulthood, where it's part of the final maturation of, say, for example, the prefrontal cortex. What we do know is that obviously the endocannabinoid system during development is not static, it's quite dynamic. It's dynamic in the expression of these cannabinoid receptors, the enzymes that synthesize and break down our endogenous cannabinoid ligands, and these make a really big difference of when during development perhaps this system is perturbed to a much greater extent than what the endocannabinoid tone would normally do. It's critical also in this growing age of cannabis use that, unfortunately, during the developing brain, there's a lot of time periods of exposure to cannabis. One, as we see in the US, and these numbers are even older, that there's a growing use of cannabis during pregnancy. Many women are targeted, telling them that cannabis can alleviate their nausea during pregnancy and so on. And we know that many people especially in the first trimester, will use cannabis. But even during breastfeeding, at the time of when children are born. And this could impact on our endogenous cannabinoid system. So the question, I usually go and show some of the work that we did to get started, but a big question is, if we're talking about risk, does it really matter what happens early in life or not? And so can we look at what prenatal exposure impacts later in life? Those studies led us to our animal work, but I'm going to go back to the fact that I'm masochistic in doing these kind of studies, but so is one of my colleagues, Yoko Nomura, who's more masochistic. She was one of my trainees. And Yoko was interested in looking at prenatal stress and looking at the children after they were born and following them up across the different years. I wanted to look at cannabis. And when women are pregnant, everybody can have different stressors, but one thing became an objective stress for us that really helped us in a part. So in the US, in Northeast, in about 10 years ago, actually 2012, we had a hurricane, Superstorm Sandy. This is Manhattan underwater. And we used that actually as a common stressor that women who were pregnant at that time, well, we were studying women before that, during that time period, and after, and we took biological samples. Also, not only biological samples, we also took a lot of clinical measures as well. But I'm just going to show you a couple of things. The question was, does prenatal exposure make a difference? And so we were trying to think of how could we look at that. So we decided to look at the placenta. And the placenta, for many people, we think about as the third brain because it's the most critical temporary organ where whatever happens to the mom, also the fetal brain, but it's through the placenta that interaction occurs. And we know that it's not just critical for obviously supporting fetal development, but we know that placental programming is also linked to later psychiatric risk. So what we decided to do was to try to answer the question, does cannabis impact the prenatal, the placental environment by sequencing the placenta? And what we saw, we had our biased a priori questions in looking at endocannabinoid-related genes, for example, like the cannabinoid receptor itself. And we could see that the expression of the cannabinoid receptor correlated to the amount of cannabis the women reported using. But what we found was, in an unbiased manner, was more surprising to us. Probably it shouldn't have been in a way, understanding the endocannabinoid system in large part. But what we saw was this huge dysregulation of the immune transcriptome in the placenta. No matter how we studied it, it showed up. We also can see that there were sex differences. And it was mainly the male placenta that showed these significant alterations of the immune. Here it's in yellow are the immune-related genes. But obviously, human studies are very complex. I will tell you, as many of you know. And so what we do is that we have our animal models. Here we expose, this is with a vapor chamber, that pregnant dams get exposed to THC and a little CBD. I'm going to come back to that, well, for anybody who has questions. And we could see in the placenta of these animals the same thing. It replicated what we saw in the women with the placenta that had consumed cannabis, that there was a significant dysregulation of the immune transcriptome in the placenta, and mainly the males. And we had seen male predominant changes for a number of other biological systems before, but that was when we were studying the brain, and here it was in the placenta, and even in rodents where the sibling next to them in the same womb, the placenta differed. I'm not going to, especially in this audience, go through all the things that we saw. Like I said, we looked at a lot of biological processes, looking computational biology, and again, immune was very strong. Another thing that I'm just going to emphasize, because I'm going to come back to it later, were these epigenetic changes. Here, for example, histone H3 lysine 4 that I'm going to come back to in terms of methylation. But it's not only on the gene expression level that are changed in the placenta. We also looked at the proteins in terms of the proteome, and once again, both in the human and in the rat placenta showed these significant immune-related changes. But our question is, does it really make a difference for future phenotypes? And so, as I said, we've been following these kids from they were in utero. I'm only going to show a few things today, and we can say that when we look first, even at when they were early childhood, at their hair in terms of, well, and even many different physiological things, but here, for example, when you look at the cortisol levels, those offspring that had prenatal exposure to cannabis showed increased cortisol levels, even changes in their heart rate variability when they were stressed. But we also look at the kids, social workers, psychologists look at the kids every year. And we could see that even early on that there was a difference in the children who were exposed to cannabis prenatally, that they showed greater anxiety, greater hyperactivity and aggression. But one thing that's also clear was that there was an impact of stress. So the prenatal stress exposure exacerbated the anxiety and other measures that we had in a very profound manner. And so that even when the children, we could see the age of their first, the age of the first, I won't say diagnosis, but in terms of their anxiety and different behaviors that showed up, it was those that had both exposure to stress and cannabis where it was the greatest. So there's a synergistic interaction. But was prenatal, the placental changes relevant to anything? So what we can look at is, again, looking at the gene expression transcriptome in the placenta and see, does it predict anything? And one of the things in doing this computational co-expression gene network analysis was that the immune transcriptome actually correlated with their anxiety behavior later in life, only in the women that have been exposed to cannabis. In our new study, we also replicate the same thing, or a new analysis that is the immune gene expression in the placenta that predicted anxiety. We also see that the phospholipids, in terms of lipid processing important for endocannabinoid function, correlates more with depression scores later in life. So the human studies told us one thing. When we first started doing our animal studies, we had mainly looked at the adult brain. But the human studies told us that actually, even during infancy, you can start to see behaviors that are linked to the prenatal exposure. So now we start to go back into our animal work, where we can look at earlier time points and not wait until adulthood. So what do we see? Obviously, in a rat, it's very difficult to look at many of the clinical things that we can look at in humans, but there are certain behaviors that you can start to look at. For example, ultrasonic vocalization, in terms of that social communication with the mother, for example, early on. And we could see that those animals that were, during infancy, that were exposed prenatally to THC, they showed changes in this ultrasonic vocalization, even in the types of patterns of these calls in terms of, quote unquote, the rat language. We could also see that during another behavior, again, during infancy, that's important, like homing behavior for cognitive function, that there was a difference. But actually, when we looked at the circulating cytokine levels, it correlated to after these behavioral tests. The males show very significant differences in their cytokine circulating levels. Indeed, you can see huge differences when we're looking mechanistically. I'm not going to go into the mechanistic detail where we see where these immune changes are being mediated, but for example, NF-kappa B signaling, very different in terms of the brain, for example, accumbens gene expression levels with the circulating cytokines that differ in the animals that were exposed to THC prenatally versus the vehicle animals. We can also look into adulthood, follow them into adulthood, and we see once again, for example, social play differs significantly in those animals that were exposed prenatally to THC, but mainly the males, once again, not the females. From early on, we have been looking at prenatal exposure, like I said, and looking into adulthood from our early studies where we had adult animals self-administer heroin. Those animals that had prenatal exposure to THC self-administer more heroin. And even when you change the conditions so they take the same amount of heroin, those that had prenatal exposure were very fast to hit the level for heroin. And if they were stressed, just mild stressors, they would seek out the heroin more. We look at multiple behaviors, looking at motivated behaviors, and we see, once again, changes in it depends on the condition, but changes in motivated behaviors in the adults with prenatal THC exposure, greater depression-like phenotype, and even changes in their hedonic state, again, often when they're stressed. So for me, the question has always been, what is the underlying mechanisms that are maintaining these long-term effects from prenatal life that would increase vulnerability? So we looked at a number of mechanisms, but as I said, and well, at that time, we thought about epigenetic mechanisms. And we know that in the rats, at least, we're not changing it's not a genetic aspect. And epigenetics is not, you inherit your DNA sequence from your parents, but genes that should be turned on or turned off can be turned on based on environment that puts these epigenetic tags on DNA and change how they're controlling gene activity and obviously the downstream proteins and behavior. So we look at many epigenetic mechanisms, and you can scan and screen for many epigenetic mechanisms. But one particular epigenetic family that comes up a lot when we looked at our prenatal cannabis animals was related to this histone methyltransferase H3K4, in particular KMT2A, which is a particular histone methyltransferase that puts on these tags. And this is really critical for transcriptional memory. And we could look at the animals in the brain, for example, the nucleus accumbens, and see this epigenetic tag that's changed throughout their life, even long into adulthood, and not just on the gene expression level, also the protein level. We can show that these epigenetic tags are critical for the behavior because we can go in, at least in the animal brain, we can now virally mediate the expression of that particular epigenetic tag, and we can, for example, look at behavior. And when I showed you, like, there's changes in motivated behavior in these animals when they're adults, and we can go in, knock down that particular epigenetic tag, and normalize their behavior. We also see this not even just in adulthood, we actually see it from infancy. This is Loredana, actually a visiting student from Bologna, who was amazing, and she just came back to Italy. And so looking in the brains during infancy, we see, once again, huge relationship to this histone methylation, this H3K4. And they correlate to genes critical for synaptic plasticity in regulating that postsynaptic density glutamatergic signaling. And if we take this particular epigenetic tag and we sequence where it's binding in the genome, one of the things that was also clear was that well, there's something missing here, but one thing that was also clear was that it was binding to immune-related genes in the nucleus accumbens. On the right, it shows the normal relationship between where this epigenetic tag binds to these immune-related genes. And this is what it looks like in the accumbens of adults with prenatal THC. It's completely reorganized, and the synchronization of how these epigenetic tags are regulating these genes, and here it's these immune-related genes. We do look at many molecular, using different techniques as to where these are binding, but one of the things that's also clear is that they relate back to, once again, aspects of regulating the synaptic plasticity. So a lot of our work tells us that, yes, prenatal exposure to THC or to cannabis does have long-term effects. We're looking into a lot of the biological systems and trying to see, at least from the human perspective, what are some natural ways that we can go about into, quote-unquote, normalizing some of these behaviors, or at least mitigating some of these behaviors, because obviously we're not going to do the same things as we do in our rats there. Another developmental window that's important for future risk is obviously during adolescence. And of course, the prefrontal cortex is that last region of the brain to fully mature. And we know, again, that the endocannabinoid system is really critical in this developmental window. And at least in North America, a lot of teens, and these numbers are actually lower, a lot of teens use cannabis. And it's important for us to understand, to me, the adolescent developmental window, because most of the adults that we treat with substance use disorders, they initiated their drug use during adolescence. And we have a lot of teens that actually suffer from cannabis use disorder, and in large part due, as it's more of these very concentrated THC products are there. So I'm not going to show a lot of things about our adolescent window just because I want to end with like where we're going in part. But we have had a lot of debates as to whether or not, cannabis use during adolescence does anything. And obviously there are individual differences, but that's where, again, we can use our animal model and we can look into, for example, the prefrontal cortical cells. And we can look at what does adolescent THC exposure do later in life. And we can take these cortical cells, and we could see that in the prefrontal cortex, those animals that had adolescent exposure to THC, there's a strong structural difference in these animals. And in the cortex, these cortical cells, in terms of the branching, there's reduced complexity of these cells. And when we saw this, it was interesting because colleagues of ours who are studying stress, only stress, these branching changes replicated what they see with stress. So what we did was to go in to the cortex and pluck up these cells. We used laser at that time, picked up the cells to sequence them because we see these morphological changes. So what is happening in these cells? And what we saw was that there was this really a reorganization of the transcriptome in the prefrontal cortical cells compared to normal developing adolescent brain. The normal adolescent brain into young adulthood is really dynamic. It's beautiful. And you could see these normal developmental processes occurring. But in those animals that had adolescent THC exposure, they only shared about 5% of their transcriptome. There was this really strong reorganization. And where the reorganization occurred was perhaps not surprising because we saw these morphological changes, these dendritic changes, and that's what you see. But we also saw, once again, these very strong epigenetic alterations. And these epigenetic alterations, once again, came back to these histone methylation, this H3K4, KMT2A. We see it in a number even transgenerationally, and still trying to understand why these particular epigenetic modifications are so sensitive to this particular treatments. But when you look at the relationship between the synaptic plasticity-related genes and these epigenetic-related genes, again, this is what happens normally in the normal animal, and this, again, is a complete reorganization in those young adults with adolescent THC exposure. Interestingly, when we look in the brains of individuals who have been diagnosed with different psychiatric disorders, a lot of this matched what you see in those who are diagnosed with schizophrenia. So in our adolescent rat model, we also see that they increase in heroin self-administration when they become adults. Not all rats, just like not all humans. Behavioral traits even make a difference for the rats as well. We also see that there are significant changes in relation to dose. And as I mentioned earlier, as more and more concentrated cannabis products, cannabinoids have come on board, my postdoc at the time, Jackie Ferlin, who's now running her own lab, wanted to look into aspects of, does dose matter? And yes, it does. We see that many weeks after getting adolescent THC exposure, their stress hormone is still elevated, only in those animals that had the high-dose THC. They also, when they've isolated stress, mild stress, they will self-administer for more, they have more reward sensitivity. We also look into their decision-making. So for example, we can use decision-making studies in human cannabis users, and we can see when we look at the classic Iowa gambling task that those individuals that have a cannabis use disorder, they show much greater risky behavior. We can do the same thing in the animal models, and we can do the same type of, we call it the rat gambling task, and we see very similar that to the human cannabis users, that the adult rats with adolescent THC exposure, but only high-dose THC exposure, show the greater risky behavior. We can also re-expose them back again to THC later in life, and once again, it's those animals that had adolescent THC exposure, high dose, that showed the greater risky behavior when they're re-exposed to THC, and also greater impulsivity when re-exposed to THC. There's a lot going on in terms of neurobiologically that's underlying some of these behaviors. I'll just show one in regard to, for example, the amygdala. And of course, we all know the amygdala is very important for anxiety and stress-related behaviors, but it's also critically important also for decision-making, that many people, in terms of the emotional context as well with that. And here, we can see that when animals were stressed, the transcriptome in the amygdala is highly changed. And it's changed a lot more in those that had THC and stress combined, and the high-dose THC. And one of the things that we saw was that the changes in relation to astrocytes. So astrocytes are these glial, immune-related genes in the brain. They're very prominent in regulating synaptic function in terms of regulating glutamate and GABA transport, but they're highly implicated in oxidative stress and in inflammation. And adolescent THC exposure alters the structure of these astrocytes. Again, high-dose THC. And they correlate to their impulsivity and their decision-making behavior. So it's not just prenatal, also adolescent exposure is important for the pathways to addiction and addiction risk. But I want to end in some of the things in terms of what are we looking at for pathways out of it. And I talked about THC. And we know that cannabis is a very complex plant that contains over 100 cannabinoids that are unique to that plant. It contains other cannabinoids that are found in other plants as well. And obviously it has over 500 other chemicals. So in our human work, we talk about cannabis. And of course, cannabis and the complexity, as I said, of all the chemicals there. But we also know that cannabidiol, CBD, is another cannabinoid that we wanted to look at many, many years ago before CBD became so popular now, because I just wanted to see that we're studying THC in our animal models, but our humans were consuming the plant. And in contrast to what we saw, when we gave animals THC and they self-administered more heroin, when we gave animals CBD, they actually showed this reduction in heroin-seeking behavior. And that became a path to a huge research program that we have now. The thing that was interesting was that they showed this reduction when the cue was exposed to them. So when animals, just like humans, when they're taking a drug like heroin, the environmental cues or intrinsic cues can trigger the craving. So if you present a light cue or a tone or whatever when an animal is self-administering heroin, all you have to do is to show them the cue later, and they're getting no heroin, but they'll press the lever to try to get the heroin, as we call drug-seeking behavior. And when you show them the cue, that's what CBD was reducing. We know from studying our post-mortem human brains that of individuals who died with heroin users, that there's significant alteration in synaptic plasticity and so on, and I'll come to some of that later. And we could replicate that in our animal models. So for example, a classic glutamatergic gene is significantly altered when animals self-administer heroin, and we saw that CBD normalized that. But that was just studying a few genes. So we wanted to look at, we have these animal studies, are they meaningful for humans? So we did some small clinical trials just to see double-blinded, placebo-controlled, in abstinent individuals with opiate use disorder. We used the same condition as our rats, because back then we didn't even know dose, we didn't know timing, and so I just went with what the rats got. So sorry everyone who then copied our results, but this is what we found. So we found that when we showed cues to individuals who have opioid use disorder, and they were, this is a double-blinded, and they were shown a neutral cue or a heroin cue, and when we showed them when we unblinded, and the individuals who had gotten placebo and they saw the heroin cue, they craved, and CBD reduced that. One thing our animal models had showed us was that even weeks after their last CBD exposure, it was still reducing their cue, so we brought the people back in a week after their last CBD exposure, and it still reduced their cue-induced craving. One thing our animals didn't at that time, because we weren't studying it, that our humans told us, was that anxiety was a key thing as well. So those who had gotten placebo, they showed their anxiety went up, and CBD reduced that. And a week later, it still reduced it. We also, because subjective effects are very subjective, we also looked at some physiological measures. For example, here, this is cortisol level, and when they saw the drug cue, their cortisol levels went up, and CBD reduced that. Similarly, when they saw the drug cue and they'd gotten the CBD, CBD reduced the increase in their heart rate. So we have now moved on, and we're currently just finalizing a phase 2. I'm just showing you preliminary data from a phase 2 interim finding. And here, the question is, we do a lot of these things in the lab. What happens in the real world? So I'm just showing you a biometric, just measuring their physiological, doing physiological measures in their real world. Here, this is a much longer duration and dose of CBD. And we could see that in those individuals who have an opiate use disorder, they spend a lot of time in a stressful state, and CBD reduces that and moves them to a more normal state. This is just a graph of showing that. So this is just our preliminary interim finding, showing that CBD is indeed having an important effect. Hopefully that will relate to reducing their substance use. So we see that in our humans. So now the goal is to try to understand the biology of the neurobiology of it. And my postdoc, Alex Chisholm, could replicate, showing that CBD in our animal model does reduce, again, heroin-seeking behavior. And Jackie, because what we saw in our human studies, that anxiety, we did the same thing in having them shocked in terms of an anxiety model and showing them a cue. Here it's a lemon cue, and when they get the lemon cue, CBD in animals that have vehicle, their anxiety-like behavior is increased, and CBD reduces that. On a molecular level, one of the things that's really been fascinating to see is that, of course, genes are turned on and off with heroin-seeking behavior, and CBD reduces or normalizes those behavior. Similarly for the anxiety, CBD is flipping the molecular transcriptome. And there are some shared biological features of both of what CBD is doing, both in terms of the heroin and anxiety behavior. But I'll come back to one of the things I started earlier, the immune-related biological processes are also changed in regard to heroin and heroin-seeking behavior, and CBD will reverse or normalize those, both in not just the genes, but the regulators of those genes. And those are important targets, because when we look in the human brain, we can sequence, for example, the different parts of the striatum in human heroin users, and we see, genes turned on and off. What we do see very strongly in the brains of human heroin users are these changes related to actually neurodegenerative processes, mitochondrial dysfunction, but again, immune-related genes. And in the relationship in these between like the immune and, for example, mitochondrial changes related to neurodegenerative processes are significantly different in heroin users versus controls. But the thing that's fascinating is that there are clusters of these genes. And the question is, can we target some of these genes? And can we repurpose them? Because we look at them and see, are there drugs that could be targeted? And we take one of them, I'm not telling you what it is, we call it medication L. And indeed, giving animals this, we can reduce their heroin-seeking behavior, and we can even reduce it more under certain conditions that we're trying to tease out more. So it's possible to target these gene networks that we see dysregulated in humans that we can replicate in animals as well. Because one of the things that we also see is the epigenetic changes related to early life events and in adults with substance use disorders. When we look at the epigenome itself, for example, taking the striatum, we could see that if we do, again, in an unbiased manner, looking at the epigenome and ask where is the most significantly changed epigenetic modifications in the brains of human heroin users, a gene came up that perhaps we should have been looking into, FIN. It's a tyrosine kinase. And it actually accounted for the variance, for nearly 68% of the variance seen on the epigenetic level. It was predominantly changed, we saw this one in neurons, not in glia. And I said that we should probably have looked at it because FIN actually sits at the postsynaptic density and regulates synaptic function. We could replicate it in our animal models, not just on the epigenetic level, on the gene expression level. As a kinase, the functional activity of the active FIN was changed. And it even correlated in human heroin users to the amount of their heroin use, the years of their heroin use, I should say. But one thing fascinating about FIN, it's highly implicated in neuroinflammation, but as many of you know, it also regulates tau. It's responsible for hyperphosphorylation of tau, and we see in the brains of human heroin users that tau was upregulated, which in the beginning I thought is just because they were hitting their heads perhaps when they were intoxicated. But here we see it on an epigenetic level. We could replicate it in our animal models, and we could show that it's that the tau protein, it was very specifically changed in heroin users at that part of that site where FIN phosphorylates. So is it meaningful? It's meaningful because FIN is being developed, FIN inhibitors, for medications for other disorders. So again, can we repurpose what we see that's happening in the brain, in the human brain that we replicate in our animal models? One of them is being used for developing for Alzheimer's. And if we give, this is sarcatinib to rats that self-administered heroin, we could reduce this FIN inhibitor, reduce their heroin self-administration behavior. And we know that it was specific for the heroin because if we give it to them just in regular food, because we don't want to change every rewarding aspect for people who, if we're trying to develop treatments to impact on their reward, and we could see that it was very specific to their heroin self-administration. Interestingly, when we look in the gene expression pattern for with what we see with CBD, we see that this cluster of genes, again, that's very reflective of what we saw in the human heroin users, and that CBD is normalizing. So the key for us is using all of this information that we see from our prenatal cannabis exposure that makes them more sensitive, for example, to opioids, to our human heroin users, that we see some similar biological processes. And it's not just for perhaps cannabis and opioids. A number of these biological processes are changed for other substances. So there may be aspects of how we can translate this work in terms of novel treatments, not just for one substance. But we are trying to develop better understanding of what's happening in the human brain. So we're looking at doing neuroimaging in our individuals, our participants who have an opiate use disorder when they're given CBD versus placebo to see if we can mimic some of the things that, not mimic, whether or not we can replicate some of the things we see in our animal models. But as I said, we have been now going through phase 1, phase 2, and phase 3 trials with CBD. And I already showed you some preliminary data with that. So we hope that some of the other medications that I didn't tell you the names of that we're looking at, based on what we see on a molecular level in the brain, and even linked to this immune and neurodegenerative processes, may give us insights into, I took the thing where science meets brain health. We know that epigenetic dysregulation is really key to the synaptic plasticity-related changes, but it's more than that. It relates to some of the immune and these neurodegenerative processes that are linked to in the brains of, not just in terms of prenatal cannabis, but also in adults that have these opioid use disorders. And so can we now develop medications that indeed target epigenetic mechanisms, and even CBD that does target a number of these biological processes. When I was trained earlier, we were thinking that we would develop a medication for one receptor. But this is what we must understand. The neurobiology is such, it's not one receptor that's altered. And can we have, we would probably think of cannabidiol and other medications that we're looking at as quote unquote dirty. And there's no such thing as dirty. They're looking at different biological systems that are tweaking them, but they don't have very high side effects because they're not targeting one specific, being a very strong antagonist or a very strong agonist anywhere. And that may be one of the keys. So with that, like I said, I thank my village. They've been incredible. And both on our preclinical side and our clinical team, like I said, it's really great. So thank you very much for all their help and for the time.
Yasmin, many thanks for this fantastic start of the Congress.
Thank you.
It's fantastic, thank you. So first of all, really thank you for coming to this opening ceremony, and I really would like to wish you a fantastic meeting here in Milan. And please join us tonight also at the welcome reception, which will be held from 6 to 7:30 at the balcony, level 1. Enjoy.